morning, everyone. So it's a pleasure, and thank you for the organizer to invite me for this very nice meeting. I recently moved to the Institute of Regenerative Medicine, so I'm not now in the Institute of Functional Genomics. So what I'm interested in is to find a strategy to explore the cell rejuvenation, and I will present you some aspect of our research. Just to remind you that So just to remind you that with the age, we have an accumulation of senescent cells as presented yesterday in our tissue, not only in skin section, as presented by Sabatagala and Tuesday staining, but also in many different tissues, and often associated to age-related pathologies. And what we really believe now is that senescent cells may impair tissue homeostasis and regenerative ability in the elderly. So, just briefly, many different stress might lead to senescence, telomere attrition, but also injury in telomere, oxidative stress, DNA damage, oncogene activation, but also chromatin modification and alteration. And senescence, it's clearly a robust cell cycle arrest with the activation of two major pathways, p53 and p16 RB pathway. Senescent cell is like sort of a new engaging program because there is really specific gene expression profile and also secretory phenotype that is really deleterious for the tissue. But interestingly, if we look at chromatin organization, we can see that there is a real modification and formation of sort of dense heterochromatin foci with specific associated protein and also specific mark, which sign the type of the heterochromatin, which is facultative heterochromatin. And this chromatin is used during development to regulate gene expression. So potentially, this chromatin is reversible and reprogrammable. So just to remind the previous talk that there is in between proliferating cell and senescent cell, there is some modification of the epigenome when the cell age, as presented by DNA methylation, and not only in terminally differentiated cell, but also in NSC, as presented by Wolfgang previously. And we observe that there is modification of gene expression profile, and with the age of the donors, but also with young NSC in culture, we can recapitulate the change in gene expression, indicating that probably the stress of the culture is similar to what the cell experience in the body. And in correlation, there is this modification of DNA methylation. So when the cell age and when the cell lose the capacity, the proliferative capacity, it's probably due also to modification of the gene expression profile due to methylation or chromatin modification. So, and this is our working model. It means that when the cell experience the stress, what is the kind of the cell, differentiated cell or progenitor or also stem cell? And when the cells survive, we observe a sort of translation of the stress into the chromatin organization that impact the gene expression profile as a sort of gene expression signature of aging. And this is probably the major importance to impairing the cell plasticity and capacity in the aging. So we believe that probably we can have a sort of deprogramming of the cell in aging. And if it's the case, and if it's participate to aging, probably you can find strategy to reprogram them, and this is what we did. First, we tried to find some gene targets involved in the chromatin modulation by siRNA screen for chromatin reprogramming, and also another aspect which has been already presented, which is a reprogramming toward pluripotency. So for the first screen, we use a homemade library of 250 genes, and we use fibroblasts to induce senescence after downregulation of these genes, and the readout was the formation of the SAF, but we did also the reverse experiment and to see whether we can reverse the structuration in SAF, and this is what we did in the screening, the high content screening. So we identify one candidate gene, P300, which is an histone acetyl and when we downregulate P300, we lead the cell into senescence, as presented by the beta-galactosidase staining, but also with the presence of SAF. But interestingly, we block the cell cycle not in G1ase, but in G12. And if we inactivate the histone acetyltransferase of P300, we also mimic this induction of SAF and senescence. And interestingly, it has been also done with curcumin, which is an inhibitor of the histone acetyltransferase of P300. So what we also thought with this study that the induction of senescence by inhibition of P300 is independent of PC53 and P16. So suggesting that we have not only one way to induce senescence, and there is possibly a lot of different mechanisms depending on the pathway activated. So actually we selected different candidates for induction and for reversion, and we are focused now on histone methylase and also in methylbinding protein for reversion of this structure. And the objective is to find drugs mimicking the siRNA activity to trigger or to protect from senescence. So then we decided to go to reprogramming with this kind of strategy into IPS, and as presented before, it was a really amazing strategy using only 4 factors, and with these 4 factors you can really convert the identity of the cell into pluripotent, and with the similar features of cell renewal, indefinitely renewing, and also ability to differentiate in any kind of cell of the body. So this is iPS colony emerging from the fibrous culture, and this is also what we have to do to characterize iPS with re-expressing markers, but also cell surface marker that sign the pluripotency of the cell. We can effectively differentiate this iPS in any kind of cells if we guide them correctly, and in this way, in the retinal epithelial pigmentary cells, this year was announced the first clinical trial using iPS and for the correction of a pathology which is DMLA. So what we can do with iPS, definitely we can we can understand the human development, which is highly complicated to investigate. We can also model pathology, because if we start from a patient with a pathology, we can redifferentiate them, and we can also recapitulate the pathology, and there is actually more than 80 pathology derived from IPS. We can also use IPS from pharmacology, so we can have access to the diversity of the population, but we can also use this cell for drug screening and to eventually correct the pathology. And we can also use this cell for regenerative medicine with noetical cancer, and also no immunogenic cancer also for cell therapy. So what we did first was to start from cells from old patients and also senescent cells, try to reprogram them, and then try to look at the rejuvenation of the cell after redifferentiation to demonstrate that we can eventually use these cells for cell therapy. So when we reprogram with these 4 factors, the problem is that we have a sort of oncogenic stress, and a lot of work shown that senescence is a real barrier to reprogramming by using these 4 factors. So we decided to check this, and when we started from senescent cell, effectively we could not reprogram these cells. So the same year than the discovery of Yamanaka, another group identified another gene cocktail showed to have the same efficiency of reprogramming. So we checked this, and we started from replicative senescent cells from fibroblasts of a person of 74 years old. We keep the cell in senescence during 2 months to be sure that they are fully senescent with this S-beta-galactosidase staining, and also increase in P16, P21, and the presence of this structure of heterochromatin. When we try to reprogram them, I've presented here on this wall, there is some colony with phosphatase alkaline appearing with proliferative metasenescent cells. And when we try to improve the efficiency with modulator of the chromatin, does not work at all, and we only obtain colony with the 4 factor on proliferating cells. So we mixed the 2 cocktail, and we reprogram with 6 factors, and one initial step was the reversion of the SAF, and then restarted proliferation and IPS derivation. So after this, there was a very long characterization of the IPS, but interestingly, we have similar efficiency from with the 6 factor with proliferative of our senescent cell. So we follow the re-expression of pluripotency genes by qPCR with IPS from senescent or proliferative, re-expression of cell surface marker, and the ability to differentiate in all the three germline of the embryo with a specific marker. And then we try to reprogram very old patient, centenarian, and we observed a similar efficiency than for senescent cells. With this patient, they re-express cell surface marker and also differentiating any kind of three germline of the body. Then, if we look at, and we selected here 3 different IPS clones from 74 proliferative, senescent, and 96 years old, and we look at the re-expression of all the pluripotency markers, and then we, with transcriptome analysis, we clustered all these IPS with human embryonic stem cells and with different IPS derived with 4-factor only, and we observed that they cluster more with human embryonic stem cell than IPS with 4 factors, indicating that probably the reprogramming was better with 6 than 4. When we look at the differentiation ability with teratoma, it was as well. The clone was stable in karyotype analysis, and we also look at raising of the hallmark of senescence, which are P16 and P21, but also we measure the size of the telomeres, and when we look at telomeres in senescent cells, when we drive into iPS cells, we re-elongate the telomere. Again, for all the people, centenarians, we also re-elongate telomeres, and interestingly, in some, we have an optimized size of telomere compared to control embryonic stem cell, and it has been also confirmed by Q-fish analysis, indicating that maybe with our 6-factor we have a really nice reprogramming. And this is also with genes involving telomere metabolism. By clustering, we found also that our IPS clustered are more clustered with human embryonic stem cell line. We also look at the mitochondria activity. What we know is that during aging, there is an alteration of the mitochondria function. And if we follow the health of mitochondria with such a fluorescent dye, we can see that during senescence, if we compare the green and red fluorescence, which is a marker of healthy mitochondria in the cell, we can see that with this ratio, we can observe an alteration of the mitochondrial function. So, in our fibroblast, we have similar decrease in the ratio in senescent cells, but when we reprogram them, we reach a ratio corresponding to human embryonic stem cell, indicating that we switch the metabolism in iPS cells. We also decrease the number of mitochondria in iPS, which is a characteristic of human embryonic stem cell. And we found similar distribution of mitochondria in morphology than in human embryonic stem cells. What we did next was to redifferentiate this iPS in a fibroblast, and we restore the proliferative capacity of these cells from, for example, senescent cells. We gain, after redifferentiation, 62 possible division. But interestingly, we were able to go in the replicative senescence again, indicating that we maintain this ability, and we are not in a situation of immortalized cell. If we look at the transcriptome of redifferentiated cell compared to human embryonic stem cell redifferentiated in fibroblasts, we observe that they cluster, indicating that the gene expression profile correspond to a young cell, and it was also equivalent for genes involving metabolism. So we really demonstrate that it's possible to reprogram senescent cells or aged cells, and it's not really a limit to reprogramming. We check many different aspects, and probably it will be useful for cell therapy to use these 6 factors. and also to reprogram premature aging syndrome for eventually drug screening. And this is what we started to do with some premature aging syndrome. And thanks to a stem cell core facility we implemented it in Montpellier to produce a national pluripotent stem cell library and to favor industrial development for lipase derivatives. So next challenge, we have to improve the efficiency of reprogramming because it's a very poorly efficient process. To improve the safety also with chemicals, for example, now we can reprogram with chemical and modifying the cocktail. But what an important issue is how could we really inject the cell in the tissue to be repaired? Do we have to clear the residing senescent cell as presented before? And also there is a lot of progress to do to out-deliver the cell in the injury and or in the tissue. And what we have to know now also is if we are able to reform tissue of organs and eventually with different strategy of chimera. And for rejuvenation process, are we obliged to go to iPS? So can we just epigenetically reprogram the cell to rejuvenate them? This is, an important question. This is to thank all the members of my team who did the work and collaborators also. And probably the last slide will be to present you what is the step envisaged now to sort of the formation of organs to avoid this kind of cell therapy, because we have a lot of progress to do in any kind of cell therapy for any kind of tissue, and probably a nice way would be to eventually replace the organ and to create an organ. As you know, if you inject iPS in blastocysts of mouse, you can produce a chimera. And as you can see, you have chimera for many different tissues. But if you block, if you block one gene, one master gene, like PDX1, which is involved in the formation of the pancreas, it has been demonstrated that you can form a rat pancreas in mouse. So, is it possible to use iPS technology to produce full organ, which is probably the next step in cell therapy? Thank you for your attention.
Thank you very much. Do we have any questions? Okay, good. Oh, sir.
Hello. Just a curiosity. There are some suggestion that in aging cells there can be a reactivation of transposons that can lead to a permanent change in the genome. Have you checked if in your cell this happens, or if the cells that are able to be reprogrammed efficiently are the cells that have not a real great change in the genome by transposons?
Yes, in fact, we didn't check that, but you're right. In senescent cell, it seems that there is reactivation of transposon, but we don't know exactly what is the impact of this. And during the differentiation and development, there is some jumping of transposon, and we are already mosaic for this, and especially in the neural system. So we don't know exactly. Probably an important issue will be to really characterize and to have a good quality control of iPS cells before to use them in therapy, and maybe sequencing, full sequencing will be the best to select the good clone. It's become more cheaper and cheaper, so probably.
Thank you.
I'm sorry, there was one more question.
So thank you very much for this talk. It's impressive to see how fast it goes. In a few years, the stem cells created from old patients and making new tissues. That's just remaking some new from old. Very impressive. And so there is a question in the end. So if we go to there is one way, which is to grow organs and to graft them. And ideally also, other people are working on trying to not do that, but rather rejuvenate the body itself. Are you following this as well? I mean, I guess like trying to put the factors in the body in some way in order to locally force the body to rejuvenate, or
I mean, using these factors and maybe molecules that mimic the effect could be really beneficial, because we could with this understand how to rejuvenate the cell. It's just for instance, proof of concept. There is some mouse model with reprogramming factor in the body, but we know that it's lead to teratoma. But maybe we can understand more if we use sort of transient reprogramming, eventually in vitro, not in vivo, to try to reprogram them and not to go to this IPS stage. Because it seems that if you use this factor transiently, plus a master gene that directs the differentiation, you can eventually maintain the differentiation, but also to modify the chromatin. So probably it could be a nice strategy.
I think everyone is interested. That's why.
You are interested.
Well, maybe not. There is a leaping tongue, someone in the US who irradiated slightly. It's important, some aged mice, and injected some bone marrow from either aged mice as well or young mice, and they lived longer, especially with young bone marrow. And also they had some improved bone density. That's the thing that the researcher was looking at. So that's one example of trying to have those cells absorbed in the body. Are you working on that as well? Because you are doing so many things. So.
Yeah, it's an interesting study because we have to know in which context we could rejuvenate the cell or tissue. And this is the main problem also for cell therapy, because if we inject cell, we don't know if this cell will be able to rejuvenate the tissue. Is it necessary to have a stress before, to suppress cells, really we don't know. So in this situation, you mentioned, you effectively destroy some cell, and you let the place to replenish the tissue. And probably it's an important step before cell therapy. And accordingly, there is some strategy for skin aging, which named peeling. And peeling it uses very amazing drugs and molecules to inflammate the skin and to destroy cell, but the result is incredible, really. So it means that if you stress, you damage cell, you probably suppress cells and make the stem cell to proliferate and eventually to replenish the tissue in a more young situation. Yeah. Thank you.
Right, well, thank you very much.